this is the story that I'm going to tell you today, is how we can use our computational approach to dissect the contribution of specific molecule to the 3D genome organization. Particularly, we were interested in a specific type of RNAs, because over the year, it is known that the RNA is important in maintaining in the normal chromatin morphology. Indeed, experiment in which the cell are treated with RNase treatment for a few minutes show a completely altered nuclear structure. Interesting, there are evidence for a particular types of non-encoding RNA that are bounded to the chromatin that can sort of rescue this disruptive phenotype. Not only, over the year, there are multiple evidence that non-coding RNA have a function in structuring the genome. For example, particular types of non-coding RNA can coat chromosomal territory, and the presence of or absence of this RNA impact the chromatin compaction. This is the case of well-known cyst, and is a role the role that that these long non-coding RNAs has in the formation of the bar bodies, but also of broadly highly repetitive RNAs that seems to have a role in decompacting the chromatin, especially in active portion of the genome. Not only, RNA is also involved in dynamic nuclear processes, as for example, as other 15 that impact the replication timing and the stability of particular autosomal chromosome. The interaction between RNA and DNA within the nuclear environment can be categorized in two types. First, there is the indirect type of interaction that are mediated by protein. But what struck my attention are those interactions that are direct. So meaning that the RNA can bind directly to the double-strand DNA within the nuclear space. This type of interaction can be either disruptive, so the the DNA duplex is altered, and these are R loop where there are RNA, DNA double-stranded type of structure that are formed. But then there is a third type of interaction that occur via RNA-DNA triplex. So these interactions are particularly interesting because they do not alter the structure of the double-stranded DNA. The RNA stretch that is called triplex-forming oligo domain of the RNA can bind the DNA directly in the major groove via the formation of this Hookston base pairing, so not canonical base pairing. There are various motifs that can be formed with this type of triplex structure, but the most stable are those ones that involve a highly purine-rich type of sequence. Over the years, there have been a lot of debates if this type of structure do exist in vivo, and and they indeed do exist both in vivo than in vitro. And interesting, the triplexome does not overlap with reported loops region along the genome. Interesting also for me was that this type of triplex interaction in vivo are stabilized by the presence of the nucleosome. So we start thinking and hypothesize, can we check if via the direct RNA-DNA interaction via triple formation, RNA binds the chromatin and has a role in spatial structuring the genome as more as a global architecture factor than a local organizer. So for this, at first, we bioinformatically identify long non-coding RNA that have a high triple forming potential, that are expressed in multiple cell lines, and importantly, once we identify the triplet-forming oligo stretch or domain on the RNA, we check if this domain is not in a double-stranded secondary structure annotation, thus implying that is suggesting that is free to engage in triplex-forming triplex structure. So with this analysis, we identify 150 London coding RNA. These London coding RNA have different length and are mainly antisense or link RNAs. But with this analysis does not only allow us to identify triplet target factor domain on the RNA, we also now can identify triplet target site along the 1D genome. And here I show you an example of a particular London coding RNA or loci across chromosome 22 that are targeted by a particular type of RNA. So on the 1D genome, the triplet target site occur in a widespread fashion, but there are some loci that seems to have a higher potential of triplet forming. These loci are mainly located in intergenic or intronic region. And then we check if these highly triplex forming loci with highly triplex forming potential do colocalize in the three-dimensional space and create a 3D hotspot. So we select only two interactions that are that involve two pairs or triplet target site hotspot that show a significant frequency of contact in the IC experiment. To our surprise, this type of cluster are formed mainly by really long range and intrachromosomal interaction. So these hotspots are enriched in poised chromatin and in repeated chromatin, and especially in low complexity repeat and simple repeat, suggesting that this recognition or like interaction between the long non-coding RNA with this particular genomic loci via triplex formation can be a really widespread mechanism along the genome. We check and control if the identified cluster in the three-dimensional space were enriched or not in nucleosome, and indeed they are depleted in nucleosome-free regions that supporting so sort of help. Our results are supported by the experimental evidence in which nucleosome stabilize this type of construct. So we can also check. We check also that multiple long non-coding RNA can bind different chromosome, and all chromosomes have multiple are targeted by multiple long non-coding RNA depending of their length and size, and then we calculate this per chromosome co-localizing triplet targets potential. And this allowed us to classify the long non-coding RNA based on their chromosomal preference, binding preference, in 4 subgroups. The first group in which the long non-coding RNA seems to be more chromosomal specific. And then there is this fourth group in which the long non-coding RNA do have the potential to bind widespreadly all the chromosome genome-wide. So to test our hypothesis that these long non-coding RNA indeed are a driver in the genome organization by bridging this, by forming or promoting the formation of this three-dimensional cluster of triplet target site, we perform data-driven modeling. So in our modeling, we represent the chromatin fiber as a homogeneous beads on streak representation, and then we use as restraint the co-localizing triplet target pairs that we identified. Bear in mind that generally these pairs are really normally less than 3% of all the possible significant interaction that we can find in the high-C experiment. With only this handful of restraint, we perform our model, and we reconstruct 1,000 of models that represent our ensemble of possible conformation. And then we generate from this ensemble a contact matrix, a synthetic contact matrix that represent our cell population. And now we check and compare this synthetic matrix with the original IC experiment to understand how well I'm reconstructing the chromosome just using this handful of information. So here I show you how we rebuild the reconstruct chromosome 19 based on the colocalizing triplet target site target by this particular RNA. It is evident that the checkpoint pattern that identify in the IC the segregation of active and inactive chromatin is sort of well reproduced. Then we quantify the similarity and the reconstruction accuracy of our model using this diagonal cross correlation. And overall, this suggests that only using almost less than 3% of all the significant IC interaction are sufficient to reconstruct quite nicely the overall chromosome. But we also put some control in place. And the first control was to check for what if we are using in our reconstruction just a repetitive element, because it's known that repetitive element have been proposed that repetitive elements do have a role in 3D genome organization without an underlying biophysical mechanism so far. And it was interesting for us to see that although in this simulation we are using almost 6 times more restraint, we cannot achieve a good reconstruction as we are the simulation driven by the long non-coding RNA. We as a positive control, we looked for the reconstruction ability of CTCF mediated interaction, since CTCF is a well-known genome organizer. And yet again, the reconstruction with the driven by the long non-coding RNA are better in agreement with the experiment. We also perform modeling using random pairs of interaction that have a similar long-range distribution that the co-localizing triplet target site, but this does not lead to any checkboard pattern in the matrix. This result is not only restricted to this particular long non-coding RNA. Indeed, we perform, and for this chromosome, indeed, we perform different simulation analysis for multiple types of long non-coding RNA belonging to the subgroup number 4, that is the widespread subgroup of triplet-forming long non-coding RNA, and in all chromosomes. And overall, we can reconstruct nicely in our reconstruction are in good agreement with the experiment. Although it's quite evident that we are reconstruct quite nice, really nicely the acrocentric chromosome, while for some of the larger chromosome, our reconstruction is not optimal. Interesting as well is that a particular long non-coding RNA seems to indeed be chromosome specific, as with the restraint from a particular long non-coding RNA, we can reconstruct better certain chromosomes than others. But since we are looking at the diagonal correlation, we can also look and check at which length, genomic length, our reconstructor is optimal. So if we, for example, the contribution of CTCF is known to occur more a short range scale. And this indeed was also the case in our simulation, where we have with the CTCF driven simulation, we can reconstruct quite nicely the chromosome organization up to 250 kilobase. That is so more or less the median distance between two convergent CTCF sites. While instead, the best contribution for our long non-coding RNA is really a long-range tail, and actually is kicking in when the contribution of CTCF is fading out. This is reminiscent of what we kind of know of chromatin organization, in which there are two main mechanisms. One the loop extrusion mechanism that acts more at the short range scale, and then the segregation of active and inactive chromatin that occur at larger genomic distance, to suggesting that our long non-coding RNA do indeed have a role in this second type of mechanism. But with our model, we can also start thinking and assessing the contribution of both mechanisms and at once. And so for this, we implemented another set of simulation in which we simulate all the chromosome. Here in gray, each line here in gray is a chromosome. So I forgot to say, in which we simulate for all chromosome a combination of using a combination of restraint from the long RNA and the CTCF. And actually, we unveiled that there is a cooperative effect of these two regimes. Indeed, when, especially in certain chromosomes, the overall quality of our reconstruction increase when we use this set of restraint. As I said, this is generally, this is the increment in quality, in efficiency of reconstruction is generally for all chromosomes, but there are, yet again, we are performing much nicer in the acrocentric one compared to, for example, chromosome 2 or really long chromosomes. So since now we established that, well, it seems we established that this long non-coding RNA do have a role at long-range effect where the segregation of active and inactive chromatin occur. What we did then is checking the contribution of each of chromatin type interaction. So what we did is we decomposed the high-C experiment in homotypic or heterotypic type of interaction only, and then we check how well we can reconstruct each of these subtype of interaction. And generally, we reconstruct nicely the inactive homotypic interaction and for inactive interaction. Then checking this pattern, we can classify and we identify like three types of subgroup of chromosome. One in which we are able only to reconstruct homotypic B interaction. Another one in which to some extent we also reconstruct homotypic A interaction, but still much better the B type interaction. And then the acrocentric chromosome are in a different cluster. And here we actually reconstruct optimally, more accurately, the heterotypic interaction, not only the B-type interaction. So that means that our reconstruction ability, it's really dependent on the homotypic B type of interaction or the heterotypic interaction present in a given chromosome, but importantly is independent of the A-type interaction that are present in a chromosome. This observation allowed us, make us propose a model by which this long non-coding RNA code the inactive chromatin patches and promote the segregation in space of the varied compartment, or prevent the intermingle of inactive and inactive piece of chromatin within the nuclear space. So I show you today how we indeed unveil the role of this London coding RNA in segregating the genome in the genome in active or inactive compartment, and that this London coding RNA target sites that are can form triplex and loci that are enriched in simple and low complexity repeat. That's really suggesting a widespread mechanism along the genome, and that this type of mechanism that we identify coexist and cooperate with CTCF-mediated short-range genome organization. And importantly as well, here we develop a computational method that allowed to dissect the contribution of specific molecule to the 3D genome structure, and especially this molecule, like this London coding RNA, that are experimental challenging as due to, for example, the high repetitiveness in nature. So for this work, I have to thank the Marc Martí-Renom past and present member, but most importantly, Marco Di Stefano, Paola Soler, and Maria Martí-Renom. And I'm happy to have questions from the audience.
Thank you for your talk.
Hi.
I have a very ignorant question, forgive me, but how do you predict, how does a prediction work when you're predicting which RNAs have the capacity potentially to form these triplex structures? I missed that.
So we use this software that is called a triplexator, and that has been actually shown to have a quite robust way of predicting triplex, and it's just based on rules that we do know about triplet formation. So like following the rules of this motif, like I was telling that we looked at purine motif, but there are also other motifs. So we look at the percentage of, the software look at percentage of mismatch, percentage of base pairing present, the length. Also, the length of the triplex has an important instability, and it's shown that below 15, 19 nucleotides are highly unstable. So it's follow like certain rules that are known by mainly in vitro studies.
So experimentally, how do people detect these triplex structures?
There are available, out there, the groups use the antibodies that are able to recognize triplex. We did not have the chance to test them in the lab, though. So
thank you.
I do know that they exist, at least two types.
I have a question about, so as I understood it, you've analyzed the DNA sequence and you're proposing that has potential to form an RNA DNA structure. Is that right? So did you calibrate at all to expression levels of RNA?
No, so we filter for it. So when we select to characterize and identify these 150 London coding RNA, so first we perform the triplex analysis prediction with triplexitor, that is the software that I discussed before. Then we apply different filter, and one of which is that this London coding RNA that we identify are expressed in at least two types. One is GM12878, that is our cell of interest, and then in another cell type.
Okay.
So we filter for expression. But
you didn't actually include in your modeling like a level of each RNA?
No.
At each
No.
Could be interesting, yeah?
No, yeah, but at the end, the RNA-seq data that we have are population-based. So right. It will be highly interesting, but we don't have information yet.